So, as I mentioned in the long video that I have presented, or I'm going to present the fold changes, p-values, the basic things for differential express genes analysis, calculation, interpretation, and understanding. Whether you are using MATLAB or Python or Perl, it does not matter. But what you need to understand, please do not make these silly mistakes in order to understand or interpret and represent in visualization, that is p-values, fold changes, log to fold changes, what is the minus log 10 of fold p-values. So these are the things which you need to understand. So first thing, you need to understand what is differential express genes and what are the differential proteins, or what are the differential express biomolecules. Very similar, summarize them. So differential express genes profiling is the computational and experimental method used to identify the genes with statistical significant changes in expression levels between two or more groups. So this is what is mostly used in case of disease identification and understanding and unraveling the mechanisms. Now, you can think in the data generation, normalization, statistical analysis. Data generation, it can be generated from any platform, whether RNA-seq or NGS platform, or Oxford Nanopore or microarray platform, it irrespective. But what you need to understand, you need to normalize the data and go for the data accuracy and correction identification. Then you need to understand the statistical analysis and interpretation while you are talking. So for example, how to interpret the p-values, how to calculate the p-values, and what are the t-tests which you need to implement in different conditions, and how to calculate the fold change, and what does in exactly mean about the fold changes. So here you need to understand then the interpretation perspective, whether 0.05 is significant or higher values or lower values are. So log to fold change, a positive log to fold change indicates the upregulated genes, negative indicates the downregulated genes. P-values adjustment, multiple testing correction is applied to adjust the p-values controlling false positives. Visualization could be done by using volcano plots or MA plots. Now, pathway analysis could be performed by using your own code, or a large number of online tools are freely available for this purpose, where you can go for gene ontology term analysis and get pathway analysis. Now, applications are in diverse fields: disease research, drug discovery purpose, treatment response, environmental sciences. Another thing, what you need to understand here while you are understanding or working for differential expressed genes or proteins or any biomolecules. What are the number of sample sets and groups? What are the number of samples in each group? What is p-values and the fold changes? For example, here there are two groups. There could be difference. Then how to identify the difference? For this purpose, you need to perform the statistical significance analysis, that is p-values calculation, and then use the t-test of your choice and for the data type or analysis goal. And in this purpose, you need to understand the null hypothesis, probability, evidence against the null hypothesis, and how to interpret them, when to reject and accept, or reject or fail to reject. So compared to the significance level, and what could be the threshold level which you need to assign for p-value. Similarly, then after that, you need to understand what is permutation also, how to go for further correction of the calculated p-values, what is then FDR, why we are doing FDR. So the FDR, or false discovery rate, is the proportion of false positives among all the significant results. And why we are doing what, which method is mostly used, is Benjamin-Hochberg procedure. Of course, there are some other like Monferrini correction also, but it depends on you which one you are going to use. FDR means false positive value divided by all the significant results. Now, how to calculate the log to fold changes? First thing you need to understand, the log to fold change is not every time you can say log to fold change, unless otherwise you have converted the normal gene expression values into log2 values. You cannot say log2 fold change. Of course, you can say fold change, but you need to first know if you have converted the gene expression values into log2 scale or not. If it is calculated, then you can say log2 fold change. So fold change is calculated For example, if you have two groups, healthy and disease group, in healthy groups, you need to take the average of all the expression values for one gene in all the samples. Then similarly, you do this thing in the cancer samples or disease samples. Then divide the bigger number with the smaller numbers. If the healthy samples have higher values in gene expression values and the disease samples have lower mean values, gene expression values, then it means the genes are down regulated in this case. And if it is vice versa, then you can say in different way. So log to FC calculation can be done manually by hand also. But since there are 20,000, 40,000 genes, so you need to write an automated code in just 2 to 3 lines. So you need to take the mean values of all the expression values for all the genes in each sample, in all the samples. Then you need to divide the bigger values, mean values, with the smaller mean value. Then you need to assign whether is it up regulated or down regulated. If the control or healthy sample is higher, then it is down regulated. If the control or either target or disease samples are showing the mean values as higher, then it means it is up regulated genes. So control is higher, then it is down regulated. If the cancer or disease or target samples are showing higher mean values, then it is the upregulated gene. So in upregulated, you assign the positive value. In downregulated, you assign the negative values. Then you say log to FC is minus 2.3 or plus 2.4, something like this. So create a volcano plot for better representation for both the values. You cannot use just one value, fold change or p values. You need to show both the values, fold changes and p values. If both the things are under your threshold condition, for example, normally for p values it is less than equal to 0.05, or in fold change it is plus minus 2.0. So the fold change must be greater than or higher than equal to plus 2.0, then it is upregulated genes, or it must be less than equal to minus 2.0, then it will be downregulated genes. But fold change is adjustable. You can also say fold change threshold is plus minus 1.5 or plus minus 1.25 or maybe plus minus 1.5, depending on how much fold change difference you have among the genes. Now you need to think whether you are going to use the software or small modules, inbuilt modules for volcano plot, or you want to write your own code. It is both the things are quite easy, but please try to understand the basics, why we are taking the fold change and how we are taking them, how to interpret them. So you then after that, you go for, after you understand the p-values, FDR, fold changes, and volcano plot, then you also think about the pathway analysis. You can also compare the fold changes calculated from different for the different platforms. For example, you can compare the gene expression values in case of microarray cell platform generated or NGS platform generated, or something like Illumina platform, or maybe TCR, PCR, or real-time PCR, or something else. So you can compare them. So one thing you need to understand, if the gene is highly expressed, then it will always be highly expressed or upregulated irrespective of which platform you are using. It can never be reversed if you are changing the platform for gene expression measurement. Now, the most common software used for the differentially expressed genes analysis is DESeq2. You need to understand the major steps are data preparation, normalization, dispersion estimation, generalized linear model, or GLM fitting model. Then hypothesis testing, model testing for correction, that is FDR, then results interpretation. Why the people are using? Because it is easy to use and it has been specifically designed for count data or NGS samples. So it handles the complex experimental. These are some things which every software developer writes. So you need to understand that it has been basically designed for NGS data. Now, this is the summarized platform. If you are using the differentially expressed genes analysis by using DESeq2, then it could look like up to from here to here you have to come. Or EdgeR is also one of the tool which is used for the differential fold change calculation or p-value calculation. Then you can think about further analysis, for example, sample clustering, then multiple species network, co-expression network. Then you can think gene expression clustering. Then you can think functional term over representation, pathway enrichment analysis. Finally, you can conclude in the pictorial view, which is here. So you need to think about the p-values, fold changes, and how they are calculated, the fold changes, p-values, then what are the target of our analysis, biological functions or networks, and what else. So thank you very much. Please don't forget to subscribe my channels. So Change by Science and the Bioinformatics for All. Thank you very much. Please stay connected with us, and so that I will be updating from the coming new videos.